All right, ladies and gentlemen, so now that you've done those Punnett square problems that I encourage you to do before skipping to this part of the study guide, let's talk about Mendel's final principle. Now, we've talked about the law of dominance and recessiveness. We've talked about the principle of the principle of segregation. And now we're going to talk about Mendel's final principle, the principle of independent assortment. So Mendel came up with this principle solely because he needed to answer one final question in his research, his 14 years in the monastery garden. So when alleles are being segregated during gamete formation, during reproductive cell formation, does the segregation of one pair of alleles have any effect on the segregation of another pair? Like if we go back and think about when we drew that diagram with the alleles in meiosis and we used capital T, lowercase t, Mendel wanted to know, does the lowercase t allele affect where the capital T allele goes? Or like, does the dominant allele affect where the recessive allele goes? Can this recessive allele affect which cell that recessive allele ends up in? So Mendel wanted to figure this out in some way. So in other words, does the gene that determines if a pea plant is tall or dwarf have any effect on seed color, right? So he's going to now compare two traits, and he's going to see if being tall or dwarf makes that plant more likely to be this or that color. And that's going to tell him if there's any effect on allele segregation, segregation during gamete formation. So Mendel would design a second set of experiments to follow two different genes passed from parent to offspring. And we know when we follow one gene, we call it a monohybrid cross. Mono means one. So if we're going to follow two genes, I bet we just call it a dihybrid cross or a two-factor cross. Yep, that seems to be the case. Now, we're not going to go into depth about these problems. In fact, I'm going to tell you to skip them because we're not going to do any dihybrid Punnett squares. They're too long, they take a bunch of time, and that's more of an Excel or AP bio type deal. So let's just talk about the concept here. One parent had peas that were round and yellow, and the other parent had peas that were wrinkled and green. So there's two traits right there. And then the problem tells us the round and yellow traits are dominant. So Mendel would cross true breeding, meaning if they're allowed to self-pollinate, they will make identical copies. He crossed true breeding parents for round yellow peas with wrinkled green peas. And he wanted to see what the offspring were, and they were, or you can write it like this, You can write it capital R, right? Because they're true breeding, they have to be homozygous. So capital R for round, capital Y for yellow, two little r's for wrinkled, and two little y's for green. And he got all the F1 offspring were round yellow peas. Now this told him immediately that round is dominant to wrinkled and yellow dominant to green. So what is the genotype of all the offspring? Well, the offspring are heterozygote for both traits, right? Because if we cross the R's, you get big R, little r. And if you cross just the Y's, you get big Y, little y. So there's your genotype for all the offspring. Now that's easier said than done, right? We can make harder problems with these dihybrids looking at two traits and do 16 square Punnett squares instead of 4, but I'm not going to make you guys go through all that. And Mendel would actually allow these offspring to self-pollinate just like he did with the monohybrid cross. Remember when he was looking for that dwarf allele, he thought it had disappeared and it re-showed up in the F2. He's going to do the same thing here and see if that's the case, looking for if there's any effect between or correlation between seed shape and seed color. Okay, now I'm going to tell you from this point on, I'm going to exit the slide. We're going to take a big skip again. We're going to skip from slide 33, so wherever you're at in your study guide, let's go to the study guide. So we started here, principle of independent assortment. I'm going to have you on the left go down to the summary of Mendel's principles. Summary of Mendel's principles. So you just start here, and everything between independent assortment where we just left off and here, you're skipping. You can delete that from your study guide, please. And that includes all of this up until this slide right here. This is where we're going to pick up. So, let's summarize these three principles that we've talked about so far in this class. Mendel's principles form the basis of modern genetics. There's no arguing that. So the inheritance of traits is determined by individual factors or units that we now know today as genes, but Mendel would just call them factors or units. Let's put all of this in before we continue. Now, genes are passed from parent to offspring. Okay, always. They have to be, because they are DNA, and DNA is what makes us us, and it's what gives rise to life. Now, each gene has two or more forms. Different forms of a gene are called, that's right, alleles. Now, some alleles, according to Mendel's first law, dominant, while others are recessive. Sorry about the formatting here, folks. Recessive should be over here. And each parent passes on two alleles for a particular trait that they inherited from their parents. Now, they will pass one allele to their offspring when the alleles segregate into gametes. Okay? So we all have two alleles, but once alleles are segregated independently via independent assortment into gametes during meiosis, every gamete, sperm and egg cell, only has one allele for our traits. And again, the alleles for one trait do segregate independently of the alleles for another. There seems to be no disruption when one allele goes into a sperm or egg cell that stops another allele from entering. Okay. We're on time here. All right, let's get through this slide. So there are some exceptions to these principles. All right, not all genes show the same pattern of dominance and recessiveness that Mendel got to so closely study. For some genes, there are actually more than two alleles, which I've told you guys. Many times, traits are controlled by more than one gene, too. And if one gene has 2+ alleles and there are 10 genes controlling a trait, that can get pretty complicated. So we're actually going to take a look at some of these exceptions to Mendel's rules. So I'll see you in the next one. Look forward to it.